let's go to the inner ear. The inner ear is where the ear really starts to get complicated. Up until now, we've just been kind of funning with you a little bit. Now things get really complicated. The inner ear is this part that's in blue here. And if we crank up the power, as you know, it's full of fluid. Actually, two different kinds of fluid. One is sort of like cerebrospinal fluid that we have in our brain and what have you. The blue here is a bony material. We call it the bony labyrinth because it's very complex. It's like getting into a labyrinth. And inside, rather loosely fitting, is a membranous material, sort of like balloon rubber or something. It's a membranous stuff. And it forms little loosely fitting tubes inside of these bony tubes, at the end of which there are these little balls, and there's this big sac here. And for this we have a tube that goes out into a structure that for all the world looks like a snail. In your head right now, this whole structure is about the size of a pea. So we're talking about something fairly small. And remember the oval window we talked about where the piston of the stapes went in and out due to the sound pressure? That would be right here, the oval window. Now let's start out on the surface and look at this when it's still just covered with bone. Here's the oval window. The stapes foot plate fits in there. It's all nice and tightly sealed so nothing can escape, and yet the seal allows the movement of the piston, and that will create higher and lower pressure of the fluid inside this really complex structure. These structures over here are called the semicircular canals because they form three half circles. They're related to balance and movement. When we move our body, we can feel the direction we're moving. I'll show you how in a bit. And also we have sensory structures in here that are sensitive to the position of our body, whether we're laying down, standing up, or whether we're being pulled by gravity. Pressure that goes in here, if this is full of fluid and it's all bone, think of a soda bottle that's full of water. Can you blow air into it? You can't even get a cork into a soda bottle that's full of water, right? Because you can't compress the water. If it's full of air, you can get a cork in because you can compress the air, but you can't get a cork in a bottle that's full of water. Well, that's the problem we have here. If we were to have a piston that goes in and out, we have to have some way to relieve the pressure. So it turns out there's a second porthole down here. This is the oval window. This is the round window. And when you push in here, a little membrane covering the round window pushes out. It's as if the bottom of the soda bottle full of water had a rubber bottom. So now when you put the cork in, the rubber bottom just kind of rounds out, right? And then you can put the cork in if you had a rubber piece on the bottom of the bottle. And that's what we have here. Let's just kind of saw into this a bit. This is a difficult dissection to do in the human body because even this bone is encased inside of other bone. Some of the hardest bone in the body is Peter's bone, named after Peter. Remember, Peter is like a rock, and Petros really means a rock. So if we cut away all that bone, we get to here. And now if we cut away this bone, we can see the hollow spaces where the membranous labyrinth fits inside here. The oval window's up here someplace. And when the pressure comes in, the pressure distributes evenly throughout. It goes into this snail shell, around the corner, all the way up to here, where it gets into a second channel, comes all the way back down to here. And so the pressure in one causes this one to puff out a bit by following this incredible spiral course to the top, and then back down another separate channel out. Let's see how well that looks in a kind of an illustration. The red would be the pressure, pulses of pressure in fluid. Remember, fluid can conduct sound too, just like air, just has a different impedance. And it starts in the bottom down here, and it goes in and it goes round and round and round, all the way up to the top. And then when it gets to the top, it comes back through another chamber, all the way back and down. And notice the two chambers are separate in each turn. So the fluid in here has to get all the way up to the top to get in the chamber to come back out here. Let's just magnify that. Here the fluid goes through this chamber and goes all the way up to the top of the spiral, and then it comes back through this chamber. Now, why does it bother doing that? because we have another chamber in between these two. This one is also filled with fluid, but it's a different kind of fluid, and the two do not communicate. When the pressure is increasing in this fluid, it causes an expansion, which causes this little plate to move down, and when it comes back, you have a comparable effect on the bottom, and the net effect is this little triangular area is bouncing up and down. The picture? Now, when that hops up and down, we have a marvelous little mechanical device. Let's take a look at that, a little higher power. This plate, I have things facing just the opposite direction now. This little plate hops up and down, and when it does, there's some cells here that are in contact with a little sort of a gelatinous, like a hard jello cover, and this cover's on a hinge. Well, let's crank up the power a little bit here so you can see it. Here's the cover, and here are these cells. The cells that are important for hearing have little hairs. They're not like hairs in your head. They're like cilia in your respiratory tract. And these little cilia tuck into this gelatinous structure called the tectorial membrane. And when this thing is pushed up and down, here's the action you get. Think of your fist as being what you see in green. Think of your hand as being the tectorial membrane. You put the tectorial membrane on top of that, and as it hops up and down, there's a sliding. See if I keep my arm hinged? As I go up and down, there is a sliding on top of these hair cells. That causes the hair cells to wiggle left and right. You have 3 outer hair cells, and you have 1 inner cell. There's about 16,000 of these cells in the ear, and they each have thousands of these, or hundreds of these little hairs, and when the hairs wiggle, that's what begins to produce the electrical signal we perceive as sound. Well, we'll go from the ridiculous to the sublime here now, and look at one of these hair cells. The recent studies show that when these hair cells bend, they open ion channels that are on the top of these little hairs. And these ion channels allow ions to come in, which changes the electrical charge inside of the cell. The problem is we have to open the gate to let the ions in and then close the gate at a rate all the way up to our highest level of hearing in terms of frequency, which is about 20,000 cycles per second. So something has to open thousands and thousands, millions of these little trapdoors and close them again at the tips of all of these little hairs, and has to do so at rates up to 20,000 times a second. Now, they found that as the hairs tip, something is attached that pulls the trapdoor open, and so help me, it's a spring. It is a molecular spring. Now, let's stop right here, go to the evolutionists and say, look, never mind, we'll give you Albert Einstein's brain, anything you want, you've got it. We'll just assume evolution's done it. Would you show us how to make this molecular spring? They can open and close at 20,000 times a second on a gated ion channel to let ions in to create electrical charges that go to the brain that are interpreted as sound, that are understood as speech. It's too much for me. And then when we look in the scanning electron microscope, here are those little hairs. Here are 3 rows of the outer hair cells. Remember, there was 1 row of the inner hair cells. And the tectorial membrane has been removed. You couldn't see it. You see, we've taken that membrane off, and now we're looking at the top. And let's just crank it up and see. This is glorious. Here are the little, these are the size of cilia, like in our respiratory tree, except they're non-movable cilia. All except 1. The one out on the tip here is movable. The rest are not movable. And they all bend when this membrane brushes their tips. Let's crank it up and look at them some more. They're like organ pipes. They're all stacked up here, just perfectly in 4 rows. And there's 1 movable cilium up here at the tip, and the tips are all tucked into this gelatinous tectorial membrane, and when that shears, it bends the cilium. Oh my, you look at this and you just think, what else can the Lord do? Well, the other thing the Lord can do is give us a sense of balance. If you've ever lost your sense of balance, you know that this is important. And we have these little canals, and there's fluid in here. And the fluid and the canals are oriented in 3 different planes. 1 in this plane, 1 in is that sensed? Well, there's a little bulge here called the ampulla, and in the ampulla we have hair cells. And on top of the hair cells we have something called the cupula. It's like the tectorial membrane. And when the fluid pushes the cupula left and right, that wiggles the hair cells, that goes to the brain, and it's interpreted as, hey, somebody's spinning you this way. Then we have other little structures called the macula and saccula. They basically are sensing gravity, and on the top of them are little stones called otoconia. The little stones are embedded in this jelly-like substance we've been talking about. The tectorial membrane, the cupula, now we're looking at these little maculae. And these little stones have weight. And it's gravity pulling on these stones that moves the hair cells and tells you what direction gravity is pulling you in, so you know what's up and what's down. So the figure skater, when she spins, the cupula is being pushed to one side by the inertia of the fluid in there. And when people bend over, the relative position due to the weight of these little stones causes them to know what direction is down. You know, it's interesting, some of the other creatures out there, like lobsters, you ever eat a lobster? Their ears actually go through little holes in their exoskeleton. And this little macula that has the stones in it is down inside that hole. The problem is every time the lobster sheds its skin, you know, they do that every so often to grow, they lose the little stones. And they have no sense of balance until they put new stones into this hole again. Only it doesn't happen automatically for them. They have to actually go out and find bits of sand, stick it in holes in their ear. Now, who told them to do that? They put the little bits of sand into the hole in their ear, and the gravity causes the sand to fall to the bottom of this chamber, and there are little sensory cells down there that say, okay, the stone hits, that means gravity is down this way. But you want to have some fun with a cheap experiment. Grow a crayfish or a lobster shrimp in a tank, and when he sheds his skin, make sure there's no grit or anything in there. He isn't going to be able to find any stones. Then put some iron filings in. He'll put the iron filings in the ear, and then bring a magnet out, and he'll go upside down. I have a few minutes here. I just can't help but tell you a little bit about the seeing eye. I don't know which is the most marvelous, the hearing ear or the seeing eye. I do know the Lord has made both of them. That's a wonderful piece of equipment you got there that you're keeping with right now. There's a clear glass window here called the cornea. It's as clear as crystal. Doesn't have any blood vessels in it. Over here it has blood vessels, but the Lord knew better to put blood vessels in this area where you're looking through. And that's really part of your skin. If you come down over the forehead, up onto the eyelid, down across the eye, it's all really related to skin in the embryo, except this skin's as clear as crystal. Isn't that great? If you're going to have an eye to look, you have a window to look through. Wouldn't it be ridiculous to have a window and no eye to look through, a window in your skin? And then you see the colored part of the eye. That's the diaphragm. That's like the diaphragm on a camera. It opens and closes. It's all automatic. You don't have to set the f-stops, you know, as you look around. It's all automatic. And then in the middle is the pupil. And when you look in here at the iris and the pupil, you're really looking at someone's brain. It's part of the brain. And, you know, the Hebrews in the Old Testament knew about the pupil. They didn't call it the pupil. They called it the apple because it was the shape of the apple. And you've heard this expression, you're the apple of my eye. Isn't it great to be the apple of somebody's eye? I was the apple of my dad's eye. Well, that expression comes from the Bible. You see the Lord referring to Israel, and I believe ultimately and referring to all of us as believers and children of Israel. He says that as they were in the desert wandering, he encircled him, he instructed him, he kept him as the apple of his eye. That's where the expression comes from. It comes from God. And King David, when he was being hounded and chased, prayed that the Lord would indeed keep him as the apple of his eye. He says, keep me as the apple of your eye, hide me in the shadow of your wings. Now, why do we have the expression, keep me as the apple of your eye? The word keep here should perhaps best not merely the apple of his eye, as we use the expression, not knowing exactly what we mean, except we're very fond of something, but that he is protecting us as we might understand that we protect the apple of our eye, reflexively to the death, even the death of Jesus Christ. Well, there is a fellow by the name of Sir Duke Elder who is, I would say, Mr. I. He's written a multi-volume set of books that you can find in most medical school libraries. And Sir Stuart Duke Elder, an Englishman, wrote a book in this series called The Eye and Evolution. And in the book, he says that unless you're willing to engage in a lot of speculation and what have you, there's been a lot of thought that have been put on the subject of the origin of the eye, the vertebrate eye, with its inverted retina and everything. And he says that up to now, it's a problem that is as yet unsolved. This is in a book called The Eye and Evolution. I wouldn't have even brought this up, except he goes further than that. He says not only is it unsolved, but assuming you don't want to get involved with a lot of useless speculation, he says it seems little likelihood that we'll ever find a solution. So you got the picture? He says we don't know anything about the origin of the eye, point one. Point two, he doesn't think we're ever going to know anything about the evolution of the eye. This is amazing. Did I forget to tell you that this book is 843 pages long? 843 pages to say we know nothing, we never will know nothing about the eye. Now, I'm pretty long-winded. I'd have gotten this across 600-700 pages easy. But evolutionists are convinced that not only was the eye not created, it occurred by chance, and so having occurred by chance, it's not the least bit surprising. It's a piece of junk. Poorly made. Frank Zendler, who has a website, you can look this up for yourself. He's a retired professor like me, only he takes the evolutionary side of things, and he says that although the human eye would be a scandal if it were the result of divine deliberation, it would be a scandalous God who made such an eye. He says there is a plausible evolutionary explanation of its absurd construction. You've heard of Richard Dawkins, wrote The Blind Watchmaker. That man knows more things that aren't true than perhaps anyone alive. He says, any engineer would naturally assume that the photocells would point towards the light with their wires leading backwards towards the brain. In other words, that the light-sensitive cells would face out towards the light, not the other direction. It sounds pretty good so far to me. He would laugh at any suggestion that the photocells might point away from the light. Wow, do they point away from the light? They really do. All mammals, all vertebrates, the retina is then upside down. Years ago, I used to work with a sheet film camera. Shows you how old I am. I mean, these big old press cameras that had the bellows. You had to put the sheets of film in, load them in a holder. You did it in the dark, and it was quite easy to put the film in upside down. So the back of the film was towards the light rather than the front. And then you went out and you took all these pictures you never get again. You go in the dark and you develop them. You end up with about 200 ukulele picks. And that's about all you get out of it. The film's then upside down. Well, that does it right there. Obviously, an evolutionist wouldn't even think of studying a retina to try to understand why it might be advantageous to have the film in upside down. He can afford to just ignore it. And this leads to bad science. Because if you ignore evolution and just press right on ahead and say, if the Lord did it, I know it's got to be, there just has to be a reason. Oh, well, you find the reasons. I could do an hour on this alone. Let's just back up a bit. The eye develops as a little ball that comes off the brain. This is the brain in the embryo. We're not talking evolution now. A little bud comes out. It's hollow. The ball gets pushed in sort of like an empty basketball here. And this will form the eyeball here. The iris diaphragm will be at this edge and that edge. This will be the retina, the inner layer. And this outer layer will be the choroid, which has a lot of pigment and blood vessels in our body. And in the middle is a space. And these two stick together. And God put them together so that they would stay together. But after the fall, just like marriages and everything else, they start coming apart. We call that a detached retina. So we can see the beauty of God's creation, and right over the top of it, we see the effects of sin. It's beautiful, but something's gone wrong. It's like running a Rolls Royce out of oil. Marvelous automobile, but it's really damaged now. And what about the cornea? The cornea was really part of the skin out here. And what about the lens? It was a little bud that budded off the skin to make the lens. So the two parts are completely different developmental sources, the eye versus the lens and cornea. So there's the developing retina. This is the developing lens. Let's move on and look at both of those. First, the retina. This is all the film in the camera right here. It's called the retina, and it's drawn very thick here so you can see it, but it's much thinner than a sheet of paper. And if we look at a piece of the retina, magnify an area like you see in the blue rim, here's a picture under the microscope of the retina, and these are ganglion cells just like you'd see in the brain. The light comes in from above, and down at the bottom are the light-sensitive cells called photoreceptors, and sure enough, their photoreceptive ends are way down here. If you're the light, they're facing this way. And so the evolutionist says, forget about it. This is bad design. Don't even worry asking why this might be optimal. Now, your first clue that the evolutionists don't know exactly the proper answer here comes from the fact that there is a group of animals that have the retinas in right, according to evolutionary principles, with the cells facing to the front. And that's the cephalopods, like the octopus. And that's why, when you talk about somebody having particularly sharp vision, you say not that they're eagle-eyed, that would be a upside-down retina, you say they're octopus-eyed, right? Or did I get that wrong? No, you see, this is silly. An eagle can sit up in a tree and can see a fish just under the surface of the water a mile away with an upside-down retina. And I have never heard that, or anyone claim that an octopus has vision that comes even close to that. Now, just behind these light-sensitive cells, we have a pigment layer. And this pigment layer is just full of blood vessels. Now, that begins to explain why the retina is arranged as it is. And by the way, this retina is sensitive to a single photon of light. One photon. How much can you improve on that? You say, well, if it was put together right, and it was the right side around, maybe it would see sharper. Well, the resolution is limited by the optics, the lens and the cornea, not by the retina. The retina is better than the lens and cornea when it comes to resolution. So you can't improve on it by simply moving things around, but you can be sure there are reasons that it is the way it is. Let's look at that. Here's a drawing, makes things a little clearer. These are the light-sensitive cells, the rods and the cones. The cones see color, the others see kind of more black and white. And the ends of these cells, the ends of the photoreceptors called the outer segments, are the most metabolically active tissues in the body. They have to be replaced every 7 days. You're burning them out. And when they're replaced, they're just shed right off the end of the cell, so you've got a bunch of trash here. What do you plan on doing with that trash? You have to have cells ready to eat it up and digest it, called macrophages. And you don't want those cells in the way of your vision. So by having it backwards, as the outer segments turn over, the cells that digest them are in this next layer out here. The other advantage is that when light comes down through the retina, it's very important that the last thing it does is fire this light-sensitive part of the photoreceptor. After that, the light must be trapped in the pigment. If it were to reflect off non-pigment things and come back to the retina, you'd burn out your retinas faster than 7 days. So that's a good reason to have this pigment right here. But the best reason of all the retinas upside down is about 95% of the blood coming to the retina is in this layer back here. These are not merely blood vessels. This is a lake of blood. 5% of the blood's on top of the retina. So you actually have to look through 5% of the blood to see. How would you like to look through 95% of the blood? It's a blood sandwich because of the metabolic activity here. So having it in there backwards is optimum. You can't improve on it. In fact, to give you an idea how great it is, several years ago, a neurophysiologist published an article in Bite. He was trying to simulate what the retina is actually doing, the signal processing going on in the retina. He says to simulate 10 milliseconds, that's 10 thousandths of a second, of the complete processing of even a single nerve cell from the retina would require the solution of about 500 simultaneous nonlinear differential equations 100 times, and would take at least several minutes of processing time on a Cray supercomputer. This computer is so big and so expensive, the Cray company doesn't own one of their own computers. Government buys it. He says, keeping in mind that there are 10 million or more such cells interacting with each other in complicated ways, it would take a minimum of 100 years of Cray time to simulate what takes place in your eye many times every second. What do you figure? Chance on that one? And what about those blood vessels you're looking through? 5% are in front. That's a little worrisome, isn't it? As you look at me up here, you are looking through a skein of capillaries. When the ophthalmologist or the physician takes that scope and looks into your eye, you ever wonder what they see in there, old movies or whatever? This is what he or she sees. They see these capillaries, and you are looking right through them. And you don't see them. And the reason you don't see them is because if the retina is like the sheet of paper, and it is, it's thinner than paper, the capillaries are like fingers right over the top of it. They're making a shadow on the retina. And you should see this, but you don't, because the brain has been taught the following little principle. If it doesn't move, ignore it. And so wherever the eye looks, these vessels just go along with it, and the shadow doesn't move on the retina. You say, but wait a minute, you can see this stand here, it's not moving, how come? Or how is that so? Well, the muscles of the eye cause the eyeball to tremor at a high frequency. And so your visual field is moving on your retina all the time, and that's why you can see it. If the muscles of your eyeball were to become paralyzed, you couldn't see anything unless it moved. And this happens to some people, so we know that this is true. But the movement of the eyeball doesn't affect the capillaries because they move with the eyeball. Does that make sense? So you don't see your own blood capillaries. Now, you could see your own blood capillaries if you could make the shadow move. That brings up another cheap experiment. One of these days they have to do a book on cheap experiments. The next picture I'm going to show you is, by general agreement, the most interesting of all the pictures that I show. Each of you are going to see your own retinal blood vessels by making a little tiny hole and looking through it. And don't do this, please. I see people out there saying, is this it? No. It must be the littlest hole you can make with your finger and still see through it. You will close the other eye, look through the little hole. Now, this is the next principle. You must move the hole left and right, not front and back, left and right, and you must do so at a fast speed, like left and right. Do that while looking at the white screen up there, and you will see little grayish silver lines start to appear. These are your own retinal blood vessels. Each of you will see your own retinal blood vessels up here on the screen. Those that don't see them, what can I say? Blind as a bat. How many can see the retinal blood vessels on the screen? Isn't this great? Cheap experiment. You can do it with a pen light. You can just walk up to a white wall. And these are the muscles that move the eye, by the way. You have a muscle on the top, a muscle on the bottom, a muscle towards the nose, and a muscle away from the nose. When this muscle contracts, your eyeball looks up. When this one contracts, your eyeball looks down. This one contracts, the eyeball looks this way towards the nose, this one away. Got the picture? That's easy. But there's 6 muscles, not 4. There's one on the top that doesn't quite fit in. It has to run front to back inside the orbit, has to send its tendon through, so help me, a pulley. It's a gorgeous little pulley. I've seen it many times. You have to be careful. You just touch it, it breaks when you're dissecting the body. And the tendon goes through there, and it attaches to the eye, and when that muscle contracts, the eyeball rotates this way, and there's a muscle on the bottom. When that contracts, the eyeball rotates the other way. So our eyeballs do this. Isn't this great? As well as up and down and left. You're probably asking, why does the Lord give us eyeballs to do this? Is this so junior high boys could gross out girls in the classroom? No, unlike the muscles that move your eye up and down and left and right, you don't have control over these muscles to rotate. The Lord did not want you grossing out the girls in the classroom. What they're for is that He knows you'll be turning your head from side to side like this, possibly when you're walking. You know, your head does a little bit of weaving back and forth. And if your field of view is rotating, you'll get sick. Lord knows that. So it's all under muscular control so that as you tip your head, the eyeballs rotate to stay right side up. Now here's a cheap experiment. When we're all done here, get together with somebody at the table, look deeply into their eyes. We've had some really nice relationships grow out of this. Look into their eyes, see a little spot on the iris diaphragm, then have them tip their head, and you'll see their eyes are rotating, staying right side up. Now, don't try standing on your head. The eyes cannot do a 180. The Lord says you have no business going from shoulder to shoulder. That's it. Okay, well, there's so many wonderful things we can say about the eye. There's only one spot in the eye right here where we see things clearly. The light comes in, and just in this spot can we see things sharply. Everywhere else, it's not quite so sharp. If you look at me, I'm in focus, but everything else is out of focus. Isn't that true? We call this the fovea centralis, or the macula. And I've heard people say, well, that shows there's no crater. I mean, if there was a crater that knew what he was doing, he'd have the whole retina sharp. When you looked, everything would be in focus. Think about it. You pick up the Wall Street Journal or whatever it is you read these days, and if every single letter on both pages were all equally sharp at once, and all of them, for all practical purpose, saying, pick me to read, you couldn't concentrate. We're too limited. As I look out in the audience, I can only see one of you at a time. If I look at the whole crowd, I see you all, but you're blurred. But I can see each of you individually. The Lord knows we can only deal with people personally on an individual basis. He doesn't have a phobia. He can see you all at once. He can see you wherever you're at any time, in the mountains or in the bottom of the ocean or wherever. But we're limited. And we have a limited area of sharpness. God wants us to, yes, deal with people at large, but focus on people individually, and that's the way we see. Well, my favorite part of the eye is the lens. Here it is. The lens is rubbery. It's beautifully clear like crystal, only it's flexible. And there are cells that grow on the surface of the lens, and as they grow, they're like those cells in the canal of the ear. They slide along the surface all the way over to the equator. And at the equator, they turn into the lens. And when they do this, a little tiny dot, it'd be one of these cells, it starts elongating like pulling an antenna out of a car. One end goes way up to the center of the lens, and the other end goes all the way back here to the center of the lens like a big bow. And so each of these little box-like cells, when it turns into the eye, having passed across the front, becomes elongated to make a long prism, and these are flexible like rubber. And so the lens can focus not like a camera lens by going forward and backward. It focuses by changing the actual refraction of the lens, changing its shape. It's much faster. Hey, and it's all automatic. The Lord could have said, I don't know how much more I can do for you. I've given you a wonderful eye. I'll give you a focusing mechanism. It'll be just like binoculars. We'll put a little roller on the top of your nose, and everywhere you look, you just focus. It'll be great. You wouldn't get anything else done. But it's all automatic, and it's held in there with strings. Little tubules that are hollow. I thought that was a nice touch, a hollow tube. Just little fine things that are attached. And when they pull, they flatten the lens so you can see at a distance, and when they relax, they come up close so you can see up close. Let's just look at that lens in the microscope. Here are those little cells I was telling you about. They're like boxes, and at the edge, they turn into the lens, and then they become elongated. That's where you see these long lines. The nuclei actually die. The cell is dead inside the lens, and these dead cells are with you for the rest of your life. And since the fall, they cease to be rubbery later on. They get hard, and your lens doesn't focus anymore. That's not the way the Lord planned it and designed it. That's the way it is now. And if we take the scanning electron microscope and magnify the Lord again, that's the way these cells look inside. They look like boards in a lumber yard. They're perfectly stacked. And the reason they keep, each one of those boards would be a single cell that has lost its nucleus. It's full of a protein called crystal, and it's the most protein-rich cell in the body. It's as clear as glass, and if you crank up the